Hello everyone and welcome back to the second part of our reproductive lecture. So here in this lecture we are going to go over the leftover topics, which is a little more detail regarding the male and female reproductive structures, as well as the hormones that are being produced and the effects that each of these hormones will have. And then eventually we will discuss how production of these hormones during the fetal life will affect the nervous development. And then as always, we will conclude this lecture by having some practice questions. Alright, so first I would like to go over the male reproductive structures as well as the male hormones. Once we complete that one in details, then we move on to female reproductive structures and discuss the menstrual cycle, pregnancy, parturition, and lactation. Alright, so starting with males. So as we just discussed, there are gonads, ducts and tubes, accessory glands, as well as external genitalia that is present in both male and female structures. So here we are going in details regarding the male structures. So gonads or testes in males is responsible for production of sperm and testosterone. Now considering that the testes require temperature of 35 degrees centigrade in order to be able to produce sperm, therefore it has to be outside the body, because inside the body temperature is closer to 37 degrees C. So that's why testes are actually being suspended in a pouch of skin that is called scrotum. And so by being suspended outside the body, testes have a slightly lower temperature of 35 degrees C compared to the rest of our body. Now when the body gets too hot, that's the time that actually the cremaster muscle, so cremaster muscle is the muscle that determines this distance of the testes inside the scrotum. So when testes become too hot, the cremaster muscle will relax to allow the testes to move farther away from the body. On the other hand, when the temperature is cold, the cremaster muscle will contract, and that's the time that the testes will be pulled closer to the body in order to maintain a 35 degrees C temperature. Now each testis is divided into a series of lobules, each of which contains a tightly packed seminiferous tubules, which are the site of sperm production. And then there are two types of cells that help with this process. One is the Sertoli cells, which help to nurse or mother these cells. And these Sertoli cells are part of the seminiferous tubules. And these Sertoli cells are actually being activated by the follicular stimulating hormone. And so basically, in other words, Sertoli cells help nurture the sperm cell during the spermatogenesis process. So these, you can call them nurse cells or mother cells. Then we also have Leydig cells that are being activated by the luteinizing hormone, and these Leydig cells will produce testosterone. Next we have the ducts and tubules. The first structure that I would like to discuss is called testes, which is basically a network of ducts that help collect sperm from seminiferous tubules to epididymis. So you know how we mentioned that seminiferous tubules is the site of the sperm production, and then epididymis is the site of sperm storage. So their transfer from seminiferous tubule to epididymis is happening with the testes. And then epididymis itself, it's basically a comma-shaped organ that lies at the posterior border of the testes. So it's located posterior to testes. And it helps to store as well as helps with the maturation of the sperm. Now, sperm can remain in epididymis for as long as multiple weeks. And during this time, they're ready to either get expelled or they could also get reabsorbed by the body. Next, we are having ductus, or the other name for which is Vas Deferens, which is a long duct that connects epididymis to the urethra. All right, so Vas deferens is connecting epididymis to urethra. And then next we are having ejaculatory duct, so ejaculatory duct, which is formed from the union of the seminal vesicle ducts with vas deferens. And then from here on, we are having urethra that will extend from the urinary bladder to the tip of the penis, and that would be the terminal duct for both the reproductive as well as the urinary system. So this is the image that I've shown you in the previous lecture. So we just discussed inside the seminiferous tubules of the testes, sperm will be produced, and via the rete testes, sperm will be stored, will be moved to the epididymis, inside which they would get stored and undergo maturation. Then we have vas deferens that will transfer all the way, it's a long duct that connects epididymis all the way to the urethra. Now combination of vas deferens with the seminal vesicles will lead to the formation of the ejaculatory duct. And then next we are having urethra, which is served both for ejaculation of the sperm, and it also can be used for urination. And as we just discussed in the previous video, if you're having difficulty remembering the order of events, it's called 7 up is the memory aid. So starting with seminiferous tubules, then we are having epididymis, after that we have vas deferens, then we have the ejaculatory duct, which is from combination of seminal vesicles and vas deferens. After that we have nothing, so ignore N. Then we have urethra, and then finally we have penis. All right. Next, I would like to discuss the accessory glands. So in the previous lecture, we briefly touched upon different accessory glands, but we didn't really go into details. So here I'd like to discuss in a little more detail what's the function of every one of the accessory glands that are present in the male reproductive organs.